Hello lovers, in this video the brilliant Dr. Redbird is going to be walking you through our OCR Gateway Separate Science Higher Biology paper to our predicted paper for 2023. So if you haven't had a go at this paper already, what we want you to do is to pause after the question comes up on screen, have a go at it yourself on a bit of paper, play the video and we can walk through the answers together.
Hi everyone, and welcome to this video. We're going to go through the Separate Biology Higher Paper 2 this time. So hopefully you've done the paper, hopefully you've done paper 1 and watched the paper 1 video, and now we're on to paper 2, and hopefully you've had a go at these questions and you've either marked it and you just want to go through it and make sure that you marked it correctly, or you haven't marked it yet and you're going to mark along with me. That's great. I'm going to go through each question step by step like in the previous video. Go through the questions, kind of point out where there's clues and tips for like analysing the questions in order to figure out what to say and how to write the answers. Go through some model answers and then explain the mark schemes to you as well. And like before, we've got the grade boundaries on there. Remember, these are very rough grade boundaries, so they're not going to guarantee you the same mark in the real exam, but it gives you a good indication of maybe where you're at with your percentage. Okay, so off we go. So section A, we're going to start with our multiple choice. Question 1. What is an ecosystem? Okay, so this is something we need to know, a definition that we will have learned, but it involves the communities living in their habitat. So that all the communities that live in a specific habitat or their interactions with their habitat. So it's not just a community, it's not just the non-living environment, which is the habitat. It's not a population and its non-living environment, it's the communities. Because a population, remember, is just one species, whereas an ecosystem is loads of species, which is a community. Which of these is not an example of an abiotic factor? Okay, so abiotic means non-living, so not non-living, which means living. So which of these is a biotic factor or a living organism? So it's not temperature, not soil, pathogens. Pathogens are bacteria and fungi, microorganisms that cause disease, so they are living, so it is C. Question 3, which of these terms describes the following genotype? Capital A, little a. Okay, so we've got homozygous dominant, that would be 2 capital A's. Heterozygous is capital A, little a, so we know what the right answer is. Let's check the others. Homozygous recessive would be 2 little a's, and then heterozygous dominant isn't a thing. You can't be heterozygous dominant. So it must be B. Okay, so question 4. What type of pathogen is responsible for the plant disease powdery mildew? Okay, so this is a content question. It's kind of a fact that we just have to know. I didn't be able to eliminate a couple though, because protist and virus, there's a couple of specific examples of plant viruses, but they wouldn't form powdery mildew, which is like a white powdery substance on the outside of the leaf. A virus wouldn't do that, and neither would a protist. They have to live inside the host. So then it's got to be a bacteria or fungus. It is a fungus. Question 5. From which natural source was the chemical used to make aspirin extracted? Okay, so this is one of those sort of plant alternatives or sources of medicine from plants. So let's go through what each of these does. So foxglove, it can't be because that is used to make digitalis. Penicillium obviously makes penicillin. And then the poppy is where we get things like opiates, so morphine. So it is the willow tree is where we get aspirin from. Again, could have remembered some of the others and maybe not remembered the aspirin and worked out this way, or just known that it was aspirin. Which of these is an example of something that is recycled in an ecosystem? So basically, which of these takes part in some kind of cycling or recycling? So is there a water cycle? Yes. Is there a carbon cycle? Yes. Is there a nitrogen cycle? Yes. So it is D, all of them. Question 7. Which of these is not caused by smoking during pregnancy? Okay, so smoking during pregnancy is related to a few things. Not increased birth weight. Increased risk of miscarriage, yes. Increased risk of birth defects, yes. Increased risk of asthma, yes. Not increased birth weight, decreased birth weight. Smoking during pregnancy causes small babies. So it's A that is wrong. Question 8. Which of these is not an example of discontinuous variation? So discontinuous variation is like categoric data or discrete data. So it's groups or categories, not continuous data, so like things with a decimal point that you can have lots of places in. So sex is male or female, blood type, eye colour, they're all categories. Height is not a category, it can be continuous data, so it is D. Which of these is the most likely outcome of a genetic mutation? So something that's most likely to happen when there's a genetic mutation happening. So we've got positive effect, no effect, negative effect, or equally likely. It's no effect. So it's most likely that a mutation will have no effect on your phenotype. Sometimes it could have a positive effect, sometimes it could have a negative effect, but it's quite rare to actually have or cause a change. Most of the time, no change happens. So it is B. Question 10. Using the phylogenetic tree below, which is the closest relative of the raccoon? Okay, so the raccoon is here. So it is on the same branch. It shares a branch with the red panda. So it is the red panda because that is the closest one. Just be careful. It's not the giant panda, even though they're close at the top in terms of labels. If you look, they're not actually on the same branch. So it has to share the same branch with the thing it's closely related to. So it is the red panda. Question 11. Which of these statements about antibodies is correct? Okay, so they are found on the surface of cells. No, that's antigens. That's where they want to get you because they think you might get them confused. They bind to specific antigens. Yes, they do. So antibodies bind to antigens and that's how they work. They are produced by phagocytes. No, they're produced by lymphocytes. And they are found in vaccines. No, again, that's antigens. So it must be B because they bind to specific antigens. Question 12. Which of these is the right order of size, starting with the smallest to the largest? Okay, so we've got a fair few here. I would draw this out. So we've got a nucleus. The nucleus is in a cell. Okay, so the cell is obviously the biggest. Inside the cell is the nucleus. What's inside the nucleus is going to be the chromosomes, because they are structures that we can see. So that means that they are bigger. The chromosomes are made up of DNA. So all the DNA together makes chromosomes, so that must mean that the DNA is the next smallest. And so then the smallest is the gene, because a gene, if you remember the definition of a gene, it's a section or a short section of DNA. So if that's our order, biggest to smallest, we just then need to put it in reverse order to get smallest to largest. So we have gene, DNA, chromosome, nucleus, cell. So gene, DNA, chromosome. So already we can rule out B and D because they're starting not with gene. So gene, DNA, chromosome, cell, nucleus. So that's wrong. A is wrong because cell is before nucleus and we know that the nucleus is inside the cell, so it's the other way around. So it's gene, DNA, chromosome, nucleus, cell. So it must be C. I would always do something like this for these kind of largest to smallest things. Really think about what goes inside what, or draw a little diagram if it's going to help you do that. It's worth it so that we don't get in a pickle and accidentally pick something that's not right. Question 13. Which of these is not a feature of a malignant tumour? Okay, so malignant is a cancerous tumour. Always remember that mal on the front means bad. In French or Latin or whatever. So it's the bad one, so it's the cancerous one. So are they fast growing? Yes. Are they cancerous? Yes. Can they spread to other parts of the body? Yes. Can they grow within a membrane and therefore be easily removed? No, that's a benign tumour, so that's a non-cancerous one. Therefore it is D. Which of these describes the products of meiosis? Okay, so products means like what do you get at the end? Meiosis is obviously the type of cell division that produces gametes. So hopefully we know that if we're producing gametes, we're looking for haploid cells, not diploid cells. And at the end of meiosis, we actually get 4 haploid cells because mitosis is 2 diploid cells. So 4 haploid daughter cells, yeah. So it is C. Okay, our last multiple choice question 15. A student placed a 1 metre quadrat down to sample the number of flowers in a field. The total area of the field is 80 metres squared. From just this quadrat, what would the estimated number of flowers in the field be? Okay, so we need to do 2 things here. We need to count the number of flowers in this quadrat, and then we're going to need to multiply whatever the number is that we've got by 80, because if this is representative of 1 metre squared, we need to see what is representative if this was multiplied up by 80 to get 80 metres squared. So those 2 are both within the quadrat, that flower is not in the quadrat, that flower is not in the quadrat, and so there's number 3. So it's about knowing whether they're more than half in the quadrat or not in order to be counted, and neither of those 2 are. So that means we have 6 flowers in the quadrat. 6 multiplied by 80 gets us 480.
Okay, so first question. The image shows 3 dogs. They are all members of the same species. What is the definition of a species? So this is a 2-mark question. It's just a definition question. Hopefully you know the definition of a species, but just to be clear that we've got to have enough detail in there to get the 2 marks. So we need to say that they're similar organisms and that they can breed, but we need to make sure we also say they can breed to produce fertile offspring. Because remember, things like lions and tigers are similar. They can breed, but their offspring are not fertile, which is why
lions and tigers are therefore different species. So a group of similar organisms that can breed to produce fertile offspring. The table below shows the classification for dogs. Complete the table. Okay, so we've got a classification table or a taxonomy table, and we need to fill in those gaps for the names of the groupings. So hopefully you can try and remember the sequence. I like to use king, prawn, curry, or fat, greasy sausages as a way of remembering the letters in order. And then we just need to remember what those letters stand for. So the K is for kingdom, the O is for order, and the S is for species. And then obviously we have phylum, class, family, and genus already done for us. So we just need to make sure we've put the right words in the right order. So yeah, use that and like an analogy to kind of remember the K, P, C, O, F, G, S. Just remember the right order. And then it's just about remembering what those letters stand for. Give the binomial name for dogs. Use the table above. So hopefully we remember the binomial name means two name or two-part name. And it comes from using the genus and the species names from the table or from their classification. And so the binomial name for dogs, its Latin name, is going to be Canis familiaris. And we need to copy it exactly as it is in the table. So capital C for Canis and a lowercase f for familiaris, because you always leave the species name as a lowercase. Okay, part D. Give one reason why the binomial name is used instead of conventional names and why they're used by scientists. Well, the binomial name is that Latin name that we just looked at from the classification, and then a conventional name would be kind of dog, for example. The first thing that I kind of think about with this question is, well, if we didn't use this Latin name, if all of the scientists around the world didn't use this Latin name and we were using the conventional names, they would be different in every language. So think like dog, chien in French. It's different in every language. So you would have to understand what was meant by that word in the language with the person you were speaking to. But if we all use this one Latin name, there's no confusion. It doesn't matter where you are around the world. And also, the fact that we've got two parts to it, we've got the genus and the species. The genus can actually tell you the evolutionary relationships of that animal. So if I know something else, like the wolf, for example, has the genus Canis, then I know that they're very closely related to each other. And that can be helpful when you're kind of looking at different species or species you're not sure about or something you don't know about. Whereas if someone just says dog, that can mean specific things to specific people. I would go with it the same name gets used by everyone all over the world, regardless of language, and it allows us to all be talking about the same organism. When the new 3-domain system was introduced, dogs were classified into the Eukaryota. Explain how the 3-domain system was developed. So it says explain how. So we need reasons that how it was developed. So how was it changed? Why did it come about? So new technology, basically things we didn't have before when the first classification system was thought up. We have DNA, we have electron microscopes, we have biochemistry evaluations, which we didn't have back in the days before DNA analysis existed. And how was it developed? So what did that extra technology allow us to do? Well, it allowed us to split the prokaryotes into bacteria and archaea, because we found out that they were actually significantly different from each other. It can't just all be lumped in together as prokaryotes. They had to be in their own separate domains. And obviously the bacteria contains the normal, true bacteria, and then we have the archaea, which are the extremophiles. So I'm going to use all this information to get my 3 marks for this question.
So this is the mark scheme for question 16. So for the first one, we need similar organisms that can breed together to produce fertile offspring. So we've got 1 mark for similar organisms that can breed, and the second mark is for produce fertile offspring. Can't say mating instead of breed. You can't say they just have the same number of genes, because that isn't the same as saying they are similar. You can say they have the same number of chromosomes. That is a way of saying that they are similar. But you need this idea that they're similar, or they have the same number of chromosomes, and they can breed to produce fertile offspring. We've got our table filling in, so we need our kingdom, order, and species. They have to go in that box. So you get 2 marks if you got all 3 correct, and you got 1 mark if you got 1 correct. So if you got kingdom, order, and species, you get 2 marks. If you got any one of those right, but the other 2 wrong, then you can still have 1 mark. Be careful with writing out the Latin, the binomial name. The first letter must be capitalised, and then no capital for the second word. So the genus should be capitalised and the species name should have a lowercase letter. So just be mindful that you've done that, otherwise you can't have a mark. You are just copying, so copying from the table should be easy. Just be accurate when you're doing it. For D, you've got reasons why we have the binomial naming system, so it's the same name used by everyone. If you just said it's the same all over the world, or it's the same regardless of language, then that's also fine, because that makes sense. It just means that it's the same word used regardless of what country you're in and regardless of what language they speak. And then the other part that's helpful is obviously the genus part gives information on ancestry or evolutionary relationships, or to know which species are closely related. That also counts and is saying the same thing there. Part E, we've got lots of things we can say here. We only need 3 marks. So the idea that the new form was, or the new sort of set of kingdoms and domains was based on DNA or on biochemical evidence. Why did we do it? Or what happened in that? Well, the prokaryotes were split into 2 groups, and then you need to say what they were split into. So they were split into the archaea and the bacteria. And then realistically, you need the description for archaea and bacteria, what they are. So primitive or simple bacteria versus the true or modern bacteria. And then obviously you've got the rest of the eukaryotes. So eukaryotes is the 3rd domain, and it includes protists, fungi, plants, and animals. If you said that eukaryotes includes organisms with cells that have a nucleus, then you can have that mark instead of going through what's in each of the eukaryotes. If you didn't get any of those marks, so if you didn't get the based on DNA, if you didn't get the prokaryotes to split up, if you didn't get the mark for saying what archaea are, or the mark for saying what true bacteria are, or a mark talking about what's in the eukaryotes, if you mentioned just the 3 domains or 3 correct descriptions of the domains, you can have 1 mark if you didn't get anything else from the list on the left-hand side.
Okay. Features of plants have been changed over many years as farmers have bred them for specific characteristics. An example is broccoli, which is the result of hundreds of years of breeding wild cabbage plants. So this is clearly a selective breeding question. We can see it's written there. So we've got the wild cabbage that has these kind of yellow florets, big leaves, really tall, skinny stems. And then we have what broccoli looks like today, and they're the same species, they're just a different variety. So over the time, we've changed them through selective breeding, but it's still the same species, it still has the same genes. So the parts of the broccoli that we eat are large flower buds with thick stems. The original wild cabbage plants had small flower buds, thin stems, and large leaves. Describe the process of selective breeding that farmers would have used to produce modern broccoli plants from the original wild cabbage plants. Okay, well, that is interesting. So that first part of that paragraph there is telling us the characteristics that have been selected for, the desired characteristics that we had clearly selected for, that have changed the broccoli over time to have these thick stems and large flower buds, which is now what we eat. So because they've given us that information, we're going to need to make sure we use it in the question. So when we say select the desired characteristics, it's told us what they are. So we should try and use them in our answer. So let's just briefly recap the steps of selective breeding that we would need to write down for any selective breeding question, not just this one. And then we can have a think about how we're going to turn that into sentences. So this is what I do for any long answer question. It's a 4-mark question. Anything where you've got a process and it's outlining steps in order, make sure you write these down, do a little brain dump before you write out the actual answer. So select the characteristics that you want, breed the animals together, select the offspring of that cross that have the characteristics that you want, breed those together, repeat over generations, okay? Because it takes a long time to do this. It doesn't just happen the first time. We've said this took hundreds of years. So we need to make sure we say about repeating as well there. So that's the kind of backbone of our answer. Now we just have to turn it into sentences that make sense for this question. Okay, so I've put choose wild cabbage plants with the thickest stems and the largest buds, breed them together, choose from the offspring that also have thick stems and large buds, and repeat this process until all of the offspring have the characteristics that we want, i.e. thick stems and large buds. On to part B. Name 2 other desired characteristics in plants that have been selectively bred. that they have been selectively bred for. Okay, so this is kind of moving away from broccoli now, moving away from kind of what they were talking about there. We're thinking about specific things or features that we should know or we have learned that have been selectively bred for in plants. So one of them is resistance to either disease or pests, and one of them is like large or unusual or very heavily scented flowers. One being useful and helpful for like crop plants, and one for more aesthetic reasons for flowers that we'd like to look at. So some major crop plants which have been highly selected for are at risk of being lost to diseases. Explain why. So this is saying, give me a reason why selective breeding can mean that some crop plants can be susceptible to new diseases. So obviously if we've selectively bred them to be resistant to one specific disease, they will be resistant to that. But this is more about kind of new diseases that crop up, new pathogens. So why might selective breeding cause this to happen? Well, if you selectively breed over and over and over again, many generations, then you're going to have lack of genetic variation because you're trying to keep the same characteristics and you're not mixing as much. So your genetic variation or genetic diversity is going to be low in the population, which means there's less chance that they'll have resistance genes that mean that they can be able to survive a new pathogen or disease. So these crop plants have low genetic variation, so they are less likely to be resistant to some new diseases or some diseases. That's all we need to say.
Okay, so the mark scheme for question 17. You've got the kind of steps for selective breeding. There's just some alternative wording on the right, but instead of breeding, you can say cross-pollinate. You can say, so choose the plants with the best desired characteristics from the offspring, instead of being specific and saying thick stems and largest flower buds. If you just said the best or desired characteristics for point 3, that's also fine. Bred repeatedly until over many generations, until all the plants have the desired characteristics. Or you could say bred repeatedly until they bred true, which just is the same shorthand way of saying that, or until they produced modern day broccoli. That's also fine for step 4. So there's lots of kind of steps in the process here that you can kind of have alternative wording for, but hopefully this is something you've learned as a process and the language that you've learned is just easy to replicate here. If you didn't say large unusual flowers for one of the things that plants have been bred for, you could have heavily scented flowers, so ones that really smell really strong. Or if you didn't put disease resistance but you put pest resistance or insect resistance, that's also fine. For 17C, you have to say genetic variation. It can be reduced, it can be low, it can be less, but you have to say the word genetic before variation to get the mark for the first one. And then less likely to be resistant, and it can be to new pathogens, diseases, whichever one you chose, but you have to say genetic variation there.
Question 18. Coronary heart disease is a non-communicable disease that kills 17 million people each year. State 2 ways in which cardiovascular disease can be treated.
Okay, so coronary heart disease, also shortened to CHD, and we have to think about how we're going to treat it. So what we're going to do to treat people who have this, and we need 3 different things. So it's caused obviously by cholesterol buildup in the arteries, the coronary arteries. So we have to think about things like drugs, like statins that we'd use to lower the cholesterol, stents, which are those little mesh sort of balloons that expand and hold them open. And then also we could replace things, like we could give new valves, new sort of bypass, a whole new heart transplant. All of these are different methods that we can use for treatment. So we just need to sort of give a description of each of the 3.
Or you can talk about lifestyle changes, obviously, so reduce or increased exercise. So we just need to pick 2 of these. So I'm going to go for statins and stents, and we'll look in the mark scheme about all the different options you can have as well.
Okay, the next one is part B is just define the term non-communicable. Break that down, it's not communicated. So remember we're talking about disease here. So we're talking about non-communicable disease in the previous question. So it's meaning in that context. Hopefully this is the definition that we've learned, so we can just repeat it. It cannot be passed from one organism to another. So it can't be spread or passed from one organism to another. You can also say obviously person to person as well. But remember, some of the diseases we talk about, incommunicable diseases, go between animals and humans as well, or plants and animals. So describe how coronary heart disease develops and affects the heart. So again, we've got to describe how, which is steps in a process. So we're going to talk about how coronary heart disease develops, what are the steps in this process, and ultimately what it causes. How does it affect the heart? What does it do to the heart? And remember, it's coronary heart disease, and obviously coronary means it happens in the coronary arteries. So with any of these long answers, this is a 5-mark question, I would make sure I always sort of jot down a little flow diagram, a little plan for myself to make sure I'm going to cover all of the steps in the process or all of the things I need to say to get these marks. So let's think about where we start. So we start with fat builds up in the coronary arteries. We have to say that it's coronary heart disease. It's not just heart disease. That's going to reduce the blood flow or make the arteries narrower. So if we have less blood flow, we've got less oxygen flowing. And where does the blood in the coronary arteries go? It goes to the heart muscle. And we have to say muscle because obviously lots of blood returns to the heart and vessels, but this is going specifically to feed the heart muscle cells. And so if we don't get enough oxygen there, what happens? Well, we get a heart attack because the heart muscle stops contracting because it doesn't have enough oxygen to carry out respiration. So this is our process. These are our steps in our process describing how coronary heart disease develops and what it causes to happen in the heart, and ultimately that is a heart attack or heart failure. So we just need to write that out as sentences. So once we've got that little flow diagram, that little bullet point, then we can just write that out. So fatty deposits build up in the coronary arteries. This narrows them, reducing blood flow and oxygen transport to the heart muscle. This can stop the heart muscle contracting, which causes a heart attack. That's it, and that should be all 5 of those marks. Okay, on to part D. So this is a graph question. The following graph shows the trends in deaths from coronary heart disease. Describe the change that can be seen from 2000 to 2018. So when it's talking about trends in graphs, what it's talking about is patterns. So just like when you did maths and talked about graphs in Key Stage 3, when you talked about it increases and then it decreases, that's what it's really talking about. So what's happening, what are the trends, what are the patterns we can see? And it says describe the change. So we're specifically talking about differences, changes from 2000 to 2018. And that's the whole graph. It's always worth double checking because it could just be asking you to talk about a section of the graph. But in this case, it's not. It wants you to talk about the whole graph. So there's some things we need to kind of think about when we describe graphs. It's different when we're talking about explaining graphs, and that's something different. This is just a describe question. So we're just thinking about how to describe the patterns we're talking about. So mostly it's say what you see. Is it increasing? Is it decreasing? Does it increase then decrease? Does it then increase again? When does that happen? Overall trends are always a good thing to start with. So you look at the graph and what's the overall pattern? Is it increasing overall? Is it decreasing overall? Then you can start to break down and look at some of the more specific differences. We need to try and use data points to back up our answers. So bits from the graph that we can talk about reading off the axes. And then obviously making sure that we've got enough different points for the number of marks we have in total. So this is a 4-mark question, so we're looking for lots of different things we can say. So let's start off with our overall trend. Overall, from the start to the end, there's a downwards trend. So there's a decrease overall in the number of deaths from coronary heart disease from the start to the finish. And you can look and calculate those numbers. So we've got sort of over 1,500 and we've got over 1,300 to start with. And then by the end, they're both under 1,000. So that's clearly an overall decrease in the number of deaths. So that's a good thing we can say. There is not just a downward slope the whole time though, because we have like an initial increase up to 2003, and then we have a decrease. So that's a bit more of a specific description of the pattern of the trends that we can see. So it increased until 2003, and then it decreased, even though overall we have a decrease from start to finish. And then we've got this difference that we can see that we've got between the two lines, that men always had a higher number of deaths than women. So that line for men is always higher than women the whole way down. If at any point it swapped or it changed, that would be another thing we could talk about. So men was higher than women until a certain point and then it changed. But in this case, it stays the same throughout. So we've got, I think, 4 things we can say. We talk about our overall trend. We look into the specifics of the increase and then the decrease. We've got this idea of using our data points to talk about the actual decrease overall, so the numbers that we can talk about. And then we've got the difference between men and women. That's 4 different things that we can talk about and patterns and trends that we can see in this graph. So that's going to get us our 4 marks. So overall, the deaths from CHD have decreased from 2000 to 2018. And then I'm going to use my numbers here. So from over 1,500 in 2003 to less than 1,000 in 2018. And I used my highest peak point, which was about 2003. But you could have used the ones at the start of 2000. That's absolutely fine. There was an increase in CHD deaths initially until 2003, and then there was a decrease. So we've described that pattern that we saw. There have been consistently more deaths from CHD in men than in women, and you can add throughout the whole time or from 2000 to 2018. So there we go, 4 points. That is question 4 done.
So the mark scheme for question 18, again, lots of options for the different things you could talk about. Lifestyle changes, so reduced fat diet, regular exercise. You need to give the idea of change. So you can't just say lifestyle, you need to say lifestyle changes or say a change. You can have statins, artificial valves, stents, transplants, any of those. Cannot be passed from one person to another or from one organism to another for the definition for non-communicable. Fatty deposits build up, you need coronary arteries. Okay, so for 18C, you can't just say inside arteries, you need to say the word coronary, because it's the coronary arteries that feed the heart muscle. And then 18D, we've got loads again of options that we can say. We said most of these. So you get 1 mark for data use anywhere in your answer. So if you've pulled a piece of data, so a bit of the axes from anywhere in your response, you can have 1 mark. And then there's 3 kind of description marks for the patterns that we saw. So deaths in men are always higher than that for women, increases then decreases gradually with time, or overall a downwards trend. So there's lots of options here that we can get marks for.
Question 19. So A is asking us to state the source of all energy in ecosystems. So that's basically asking us, where does the energy at the start of the food chain come from? So when something is asking for a source, that's what it means. Where is something originating from or coming from? Okay, so here's a food chain in case you needed a reminder. So we're not talking about the producers. We have to talk about where the producer gets their energy from. So the source of energy is light energy, which comes from the sun. So the source of the light energy is the sun. Because then if you think about it, the energy being transferred from producer to consumer is biomass. It's in the form of biomass. In the source to the producer, it's light energy, and the source is the sun. Forests are an example of an ecosystem which can contain many interconnected food chains. So there's questions about food chains. Here is an example of a food chain from an oak forest. Oak tree, moth caterpillar, blue tip. So remember the arrows show the direction of the energy transfer. And if you're not sure what a blue tip is, it's a bird. Draw and label a pyramid of biomass for this food chain. So we need to draw it, draw the boxes, and then label it with obviously the 3 levels of the food chain with 3 animals. So remember, the reason they're called pyramids is they don't actually look like a triangle shape, but we have a larger sort of bar on the bottom, getting smaller as we go up. And the producers are on the bottom because they have the greatest biomass, and we get smaller and smaller biomass as we move up the food chain. So we need 3 levels, 3 rectangles, touching each other, on top of each other, getting smaller each time, and then we need to label those with the 3 levels of the food chain, so the oak tree, the moth caterpillar, and the blue tip. The oak tree leaves contain about 1,620 kilojoules of energy. Only 10% of energy is transferred to the next trophic level. Calculate how much energy from the oak leaves can be used by the blue tips for growth. Show you're working. Okay, so this is basically, do we understand how the energy transfer works in a food chain? So if only 10% of the energy in the leaves is available to the moth, then only 10% of the moth energy is then available to the blue tips. So we just need to decrease it by 10% twice in order to get how much energy would be left over for the blue tips. So what's 10% of 1,620? 162. And then what's 10% of 162? 16.2. So we're just moving the decimal point 1 place each time. So our answer is 16.2 kilojoules. Explain what would happen to the number of blue tips in the forest if severe cold weather caused fewer moth caterpillars to hatch. Okay, so let's think about our food chain then. So we've got oak tree, moth, caterpillar, blue tip. I'm going to rewrite it because it's kind of small over there. Basically, this is just asking us what's going to happen to the numbers of blue tits in the population if the moth caterpillar suddenly decreased. So cold weather means fewer moth caterpillars hatch, so there's fewer of them that are around. What's that going to mean for our blue tit? So moth caterpillars decrease, that means there's less food for the blue tit, so they are also going to decrease. So their numbers are going to decrease or decline, and we need to say, it says explain what would happen. So stating what would happen would be just the blue tits decrease. Explaining, we need to give a reason why they decrease, or reason why those two things link together. So the blue tits will decrease because there's less food for them. And so if they get less food, they'll be getting less energy, less likely to survive and reproduce, and therefore fewer blue tits.
Finally, part E.
In some forests, this food chain can include a hawk as a tertiary consumer. Explain why many ecosystems cannot support food chains that include more than 5 trophic levels. So in this example, that would be the next level up, so it would eat the blue tit. But then the question is actually asking us why more than 5 trophic levels wouldn't happen, or is less likely to happen. So that would be another 2 levels on top. So explain why many ecosystems can't support food chains. So why can't it support that many organisms? So hopefully we're going to need to explain this. So every time we go up a trophic level, the amount of energy that passes up decreases by about 10%. So there wouldn't be enough energy by the time you get to that 5th level. Not enough energy is being passed on from the trophic level below to actually sustain that organism. They can't live because there's just not enough energy there for them to keep going from their food. So that's what we're going to say. So because or as only 10% of energy is transferred in each trophic level, there wouldn't be enough energy to sustain 5 levels, or there wouldn't be enough energy for a 5th level consumer.
So the mark scheme for question 19. We obviously have the sun, and we talked about why that's the answer for part A. It's asking for the source of the energy. So you can't just say light energy, it's where the source means, like, where is that energy coming from? So it's coming from the sun. Then we've got our pyramid. You need 3 layers that go down in size, so they get smaller each time. You need the 3 labels then for the second mark in the correct food chain order. So we need it to have the oak tree at the bottom, then the moth caterpillar, then the blue tit. It needs to look like this and have these labels in this order to get 2 marks. You have to have the producer at the bottom. The bars should be equally spaced, so sort of around the middle, and then they should be touching. Please make sure that you've not got gaps between your bars and your pyramid, because then you could lose the mark for mark 1. You've got the correct answer of 162 kilojoules for part C. If you somehow got the incorrect answer, but you knew what you needed to do in terms of working, but maybe you put it in the calculator wrong, or you wrote down the wrong units, whatever you did wrong, but you did that kind of, you wrote out that that's what you were going to do, you can have 1 mark out of 2. For part D, when saying what's going to happen to the number of blue tits, they're going to decrease, or there'd be less of them, or the population would be smaller, or the population would decline. There's obviously lots of ways of saying this, as long as you're saying the population is going to decrease in number in some way. And then you've got the because there's less food. You can't just say fewer moth caterpillars, you would have to say fewer moth caterpillars to eat, because they told you the moth caterpillars are going to decrease in the question. You need to make it clear that the reason the blue tits will decrease is because that means there's less food for them. And then the energy question, so why can't we have 5 trophic levels in a food chain? Why is it very, very rare? And that's because only around 10% of the biomass or energy is transferred at each level. That obviously comes from the calculation we did and that question where it told you that already. But this idea that you saying only 10% is transferred and therefore there's not enough biomass or energy for the organisms to survive or to live. That's the important part there. Question 20. Discuss the reasons for and against genetic engineering. The quality of written communication will be assessed in your answer to this question. Okay, so this is basically an evaluate question, because discuss the reasons for and against is the same as saying evaluate. So we're going to be evaluating genetic engineering. So for and against, pros and cons, why genetic engineering could be good and why genetic engineering can be bad. The quality of written communication point here means that we are going to have to discuss and explain what we're talking about. So we're not just going to say genetic engineering is good and this is the reason why, and genetic engineering could be bad and this is why, as in like just state facts. We're going to need to explain a little bit more and give a bit more kind of linking and link our ideas together and give as much detail as we can in a coherent way that makes sense. So not bullet points, proper sentences that can be read like an argument that we could then obviously get marks for. So genetic engineering, if you're not sure or can't remember, is where you add a gene from one organism into another's genome. So we cut a gene out from one species and then we put it into another. So the main pros, we're not going to spend too long thinking up loads of different pros and cons. As long as we've got enough that we can talk about and explain them, that's what's important. So crop resistance. So resistance in crops to things like pests or diseases. And that, the first explanation we can then give is if we put resistance genes into crops, then they will improve their yield, which means we get more because less of them are being eaten. Another good thing about that is that you're going to reduce pesticide use, because you don't need to spray pesticides on crops if they're already resistant to the pests. That helps with reducing the impact of that on biodiversity and also things like pollution. Improving yield, why is that a good thing? Well, we're making more food and there's a food shortage, or there is food security issues around the world, so that's going to help with that problem. We can make insulin, that's an obvious one, helping people with diabetes. It's a medication. The pros of genetic engineering is we can do all of this a lot faster than we would if we were doing it through selective breeding. So it's much harder to do that with crops, or takes a longer time. The cons, GM crops could be harmful or they could cause allergic reactions, which is normally what we say, because we don't actually think that most genetically modified plants are going to be harmful for humans to eat, but they could cause allergic reactions. We admit that that's something we don't know. We don't know how they're going to interact with the wild population. So this is another kind of con or a negative of genetic engineering, is thinking about what impact is it going to have on the other plant species and insects and other animal species around it. So could it be that eating it could be bad for insects or other things in the food chain? So will it affect and disrupt biodiversity in that way? And that's one of the main kind of downsides of that. We don't know what it could do to the biodiversity, because if those genes get out into the wild, so if it breeds with other wild populations, what could that cause? Okay, so here's my long paragraph, which I've written in a style that is kind of talking about the pros and then the cons separately as paragraphs, but focusing on kind of explaining and linking those points together. So genetic engineering is a faster process than selective breeding and could be used to increase the yield of important economical crops. By making crops resistant to pests, the yield can be increased, which will mean greater food security around the world. It could also reduce pesticide use, which will have a positive impact on biodiversity and reducing pollution, especially water pollution. So I've talked about some pros and then I've explained them. So what will that impact of that pro have? So just from thinking about changing the pesticide or disease resistance, we've got a lot of pros linked onto that, like we said. Other uses of genetic engineering or genetic modification include production of useful medical compounds such as insulin, which continues to save lives of type 1 diabetics. The issues with genetic engineering are that it is unknown if genetically modified crops could be harmful to humans. They could cause allergic reactions, for example. There is also a risk that the GM crops could affect other wildlife and disrupt food chains or ecosystems. This could be more damaging if the genes from these GM crops were to spread into wild populations through sexual reproduction. For example, making herbicide or pesticide resistant plants of weed species which cannot then be controlled. So if you've got crop plants that are disease resistant or pesticide resistant, that's great because they will not need pesticides sprayed on them or herbicides sprayed on them. If you create those crops and then they breed with wild species that you would normally spray to get rid of them, then if you don't do that, or if they have this resistance genes, you can't kill them, you can't control them if they are resistant to the herbicide that normally kills them and gets rid of the weeds. So they will then compete with the crops, and then we're back to square one in terms of how to reduce our yield. So this is one of those ones where there's loads of things that you can say. It's about linking them together, writing them out structurally and properly so that it reads well, and you've covered a lot of the pros and cons whilst being able to explain why they are an advantage or a disadvantage. Question 20 then, there's obviously a lot here for the mark scheme. So you've got this idea of indicative scientific points. So there's points for and points against. So we've got some of the ones we've mentioned. So points for, like improving the yield and providing pest resistance, creating insulin, faster than selective breeding, and then some against, like it's not ethical, people don't believe it's okay to interfere with nature. The seeds could be more expensive, they could be harmful, they could cause allergic reactions, it could be toxic, we don't know if it'll transfer of genes, could cause other species to have an impact, could affect food chains, food webs, etc. So all of that are the for and against points that gets you into level 2. 1 to 2. And then to get into level 3, which is the 5 to 6 marks, what we need to think about is the kind of explanations, the scientific explanations, which is this part here. So this is for level 3. So you need to have given some fors and againsts to get into level 1 to 2. And then to get up from that to level 3, you need to have thought about some of these explanations. So like we said, thinking about food security or curing hunger or malnutrition, thinking about golden rice, for example. Thinking about the fact that we're reducing pollution, like we said about the environmental causes of using pesticides and herbicides. The idea of this costing idea of companies having a monopoly on producing seeds and then they make them sterile, so you can't breed from those seeds, so farmers have to buy them every year, which means they're obviously very expensive, and developing countries, which might most need these special resistant crops, can't actually get them because they can't afford them. And that's obviously quite a complex point. It may have been something you've discussed in lessons, it may have been sort of something you've seen in other example questions or exam questions, but this is kind of, any of these, you didn't have to have all of them, but any of them show this next level of kind of linking and understanding. So if we can improve yield, how does that impact people? If we can get pesticide, herbicide resistance, how does that, what other added benefits does that have? Or the opposite, if people control this genetic modification technology, how is that a problem? Do we get a monopoly? Do we get more expensive things and companies charging what they like for it because only they produce it? And these kind of issues. So you need to have a read of your answer, and then we need to think about a couple of things. How much indicative content sort of level 1 to 2 did we include? So to get into level 1, the minimum you need is one argument for or against. To get into level 2, you needed one argument for and one argument against, and you need at least one example of genetic engineering. So an example can include disease-resistant crops, creating insulin, pesticide-resistant crops, whichever example, sterilised insects, whichever example you use, you needed an example in order to get into level 2. And then to level 3, you've got at least one argument for and one argument against. It's assumed you'll obviously have included an example, because you're going to need to talk about an example of genetic engineering in order to give the next part, which is one explanation of the problems or benefit, which is something from this level 3 category selection. So an explanation of how that method of genetic modification can actually link or improve the for, or could make the against argument worse. Okay?
So on to question 21. The diagram below shows an E. coli bacterium taken using a microscope. The actual length of the bacterium is 8 micrometers. Okay, so the first question asks us, what type of microscope was used to obtain the image shown in the diagram above, and give one piece of evidence to support your answer. Okay, so this is asking us what type of microscope. So our choices are light microscope or electron microscope. So it's clearly an electron microscope image. And this is not just because it's black and grey, although sometimes that's useful and helpful in order to help you recognise when it's electron microscope, but it tells you that this whole image is only 8 micrometers long. It's a bacterium. We can see a lot of detail here. We can see internal structures inside. So we know that this level of detail would not be achieved with a light microscope. It's too small for a light microscope to be able to show this much detail. So that's kind of both bits of our answer actually. So we've got our electron microscope, which is our type of microscope. And then I would say is that you can see detail inside the cell that is too small to be seen with a light microscope. Or the whole bacterium would be too small to be seen with a light microscope. So that's my evidence. So part B, name the process by which E. coli bacteria divide. Okay, so this is just a question that's asking how do bacteria divide? What's the name of the process? Hopefully we know that. It's just a fact we have to know. It's binary fission.
On to part C.
A student set up a practical to investigate the effects of antiseptics on bacterial growth. They placed paper discs that had been soaked in antiseptics onto a agar plate that was covered in bacteria. Bacterial plate is shown in the image below. Now, it doesn't matter that we haven't necessarily seen this before or looked at one of these before. We just see what the question is asking us first. So the bacterial plate is incubated at 25 degrees C for 36 hours. Assume there were 1,000 bacterial cells when the plate started inoculation, when time equals 0, and the time it takes for a bacterium to divide is 45 minutes. Calculate the number of bacteria at the end of 4 hours and 30 minutes. Okay, so this is just a maths question because it's asking us to calculate. So it doesn't necessarily always rely on us having to use any of our biological knowledge here. But what we do need to understand, obviously, is that when it says the bacterium divide, they divide into two. We should know that bacteria divide by binary fission, and they split into two. So that means that every time the bacteria divide, they're doubling their number. And it tells us how long it takes for that division to take place. It gives us a start number, so we're starting with 1,000. And then it wants us to know how many more bacteria will there be after 4 hours and 30 minutes of them doubling every 45 minutes. So it's just a maths question where we need to work out time, how many divisions occur in 4 hours and 30 minutes, and then work out how many doublings that is, and work out how we're going to get from 1,000, how many times do we need to double it, and that will give us our answer. So step 1 is find out how many divisions are happening. That's the first thing we need to do. So how many divisions are going to occur in the 4 hours and 30 minutes? So because it takes 45 minutes for one division, the easiest way to work this out is to turn our 4 hours and 30 minutes into minutes. So do 4 times 60 and then add the 30 minutes. Then it's going to be an E division for us. So that gets us to 270 minutes. I'm just going to find out how many times does 45 fit into 270. So it tells us how many divisions there are going to be in our 270 minutes, or 4 hours and 30 minutes. Sometimes they'll do this in questions, so it's worth always looking at the time that you're being asked to look at and the time it's given you. They'll often want you to convert time between minutes, hours, or minutes, seconds, and sometimes it's easier to do it that way than try and work in hours and minutes. So 270 divided by 45 gives us 6. So that means in 270 minutes, they're going to divide 6 times. So now all we need to do, starting at 1,000, double that number 6 times. So 1,000 becomes 2,000, 2,000 becomes 4,000, 4,000 becomes 8,000, 8,000 becomes 16,000. So let's just check how many divisions have we done there. 1, 2, 3, 4. So I've still got 2 more divisions to go. So 16,000 doubles again to make 32,000, and that 5th doubling. And so then 32,000 becomes 64,000, and that is our 6th doubling. So make sure when you do, you're going to write it out like this, count the arrows. So the arrows, if you're writing it out like this, the arrows is the division or doubling. So our answer is 64,000.
To determine the most effective antiseptic, a student wanted to measure the clear zone around the top 2 discs on the plate. To describe how to determine the area of the clear zone. So we're just looking at those top 2 that've got that clear zone of bacteria around them, and it wants us to describe how to determine the area. So it wants like method steps, so literally methods, kind of what you would do step by step in order, and how you would measure and then calculate the area of those kind of circles that we've got. We can't just measure it once because that's not very reliable, so we need to try and make sure we measure it twice. But you measure it once, the kind of length, the diameter of that circle, and then you're going to go 90 degrees or right angles to measure it again, and then we can take a mean of those 2 numbers. So we've taken like the 2 widest parts, we've done it on 2 different angles, and then we can calculate a mean from that. So once you've got the mean diameter, we can calculate the area of a circle. Hopefully we should know that the area of a circle is pi, worked out by pi r squared from maths. If you can't remember that, I like to remember the rhyme, when you need a hole repaired, what you need is pi r squared. So a hole repaired means you need a hole, you need to find the area of the hole you need to fill in, and so you use pi r squared. And so with the diameter we can do that because you can half the diameter to get the radius. So this is a bit of a maths question as well. But realistically, as long as you've described how you would measure it, how you would calculate the mean, and then say that we'd use that mean diameter to calculate the area, that's really all we need here. So measure the diameter of the clear zone around each disc twice at right angles to each other. calculate the mean diameter for each disc, or the diameter of the clear zone for each disc, and then using the mean diameter, calculate the area using pi r squared. Okay, part E. Explain how a population of antibiotic-resistant bacteria might evolve from non-resistant bacteria. Okay, so this is an evolution question. We're explaining how something happens, which means we need the steps in a process. And we hopefully know that when we talk about evolution, we're basically just talking about change over time through natural selection. So natural selection is the process which we're going to describe as part of our answer, and that's kind of how we're going to say that they could evolve over time. So we need to think about, generically, for any evolution question, there's going to be a series of steps and points that you have to include, and then you just try to adapt your answer to make it specific to the question we're answering. So variation exists in the population, so there's differences between individuals in the population, and that's caused by mutations. So random mutations create this variation. Some with various mutations that will be helpful will survive and reproduce. That's really important. That means they pass on their genes to their offspring, and therefore the number of individuals with that adaptation or characteristic or whatever it is increases in the population, so increases in number until there's lots of them. So now we need to just take these bullet points and we're just going to write them out but make them specific to the question. So we need to talk about bacteria, we're talking about resistance. So in this case, the mutation, the variation that's caused by mutation is that some of these bacteria are going to be resistant and some won't be. So the mutation is going to give some bacteria the ability to be resistant to antibiotics. They are going to survive and reproduce. They are going to pass on those resistance genes to their offspring. So more and more and more bacteria that have the resistance genes are created compared to the ones that don't, because they will die out when they get treated with the antibiotic. So there is variation in the population of non-resistant bacteria caused by mutations. One mutation makes some bacteria resistant to the antibiotic. These individuals survive and reproduce or replicate, passing on their resistance gene. So the number of resistant bacteria increases until it's the whole population. So until you get the only ones that are left are going to be resistant.
So part F. New antibiotic drugs are being developed for use in the future. Describe the 3 stages of testing drugs. So 3 stages, 3 marks. Just make sure we get enough detail in here, and make sure you don't leave out steps in a process just because you think you've got written enough to get enough marks. So let's talk about as much detail as we can. So testing drugs is from when we start testing a drug for the very first few stages all the way through to like getting to a point where it's approved for produce and then use on patients. So we always start with cells in the lab. So it's like preclinical trials on cells or tissues. Then we upgrade that to animal testing, again still in the lab. And then we go to testing on humans. So clinical trials are what we call human trials, but normally we use healthy volunteers first, because remember we're checking to see if the drug is dangerous in humans but not sick people, and then we do testing on people with the illness to see if the drug works. So we've got to include all of that, because that's the full range from start to finish, even if we feel like that might be more than 3 marks. But I would always say go with as much detail as you can and make sure you see something through to the end if you know everything that you think you can about one topic. So preclinical trials on cells in the lab, making sure we've talked about cells and the idea of preclinical trials. Then animal testing, again, still in the lab. That still comes under preclinical trials. It's one of the stages of that. And then we go to clinical trials, human clinical trials on healthy volunteers, and then patients or sick people or people with the illness. Okay, so the mark scheme for question 21, as we said, so electron microscope, or if you were specific and put TEM, so a transmission electron microscope, that would also be correct for the type of microscope for part A. As we said, too small to be seen with a light microscope, or you can see detail inside the cell, or you need high magnification or resolution to see that detail in bacteria that obviously a light microscope doesn't have. You will not get the mark for how bacteria divide if we talk about mitosis, because that's not how they divide, it's binary fission. The marks for the calculation, for the division, you get a mark for showing that you've worked out that it should be 6 divisions. You get a mark for showing that you understand it should double each time, and then there's a mark for the answer. Obviously, if you didn't show any working and you just put the answer of 64,000, then you can get 3 marks. But if you didn't quite get 64,000 or you went wrong somewhere, some people might have accidentally miscounted and stopped at 32,000, for example. You've shown that there were 6 divisions beforehand and you showed that you understood it should double each time, or you've got evidence of that from your working, then you can have 2 out of 3. To calculate obviously that zonal area for part D, there are 3 marks on offer here. So measuring the diameter of the clear zone in 2 different places, 90 degrees to each other. So that's all 1 mark. So this idea that you've got 2 different places and that they should be 90 degrees to each other, measure that. Calculating the mean, and then using pi r squared to calculate the area. That's your 3 points there for 3 marks. Quite sort of a lot there for 3 marks, but it's a maths question. It is just, can you calculate area? And you're just describing how you would calculate area. So the really hard part is that first mark, but calculating the mean and using pi r squared, it shouldn't be that complicated. Then we've got our long answer question with the evolution question. So underlined are all the words that we absolutely have to have in our points to get the marks. So you have to say variation to get the first mark. You have to say mutation to get the second mark. You'd have to say resistant to the antibiotic, or you would have to say something like mutation makes them resistant or gives them resistance. There's got to be this idea that you have linked the idea of the mutation with them being resistant to the bacteria. Resistant to the antibiotic. And then resistant bacteria survive and reproduce. This is a phrase that we should know. It underpins the whole point of natural selection questions. So natural selection relies on 2 things: that there's differences between individuals, so that's the variation and the mutation part, and that the ones with a certain characteristic caused by that mutation or difference are going to be able to survive and reproduce, because then they can pass on those genes, those alleles, those mutations to their offspring, and therefore we get an increase in that phenotype, those adapted organisms, to the population. They increase in the population because they can survive and they can reproduce and they can pass on their genes. So this idea that you get a growing number of them increasing because they're able to survive and reproduce. And then, as we said, preclinical trials on computer models and human cells. I would say if you didn't talk about computer models, but you talked about human cells, then you can still have the mark for 0.1. But ideally, yes, you should remember that you can use computer models as, like, in terms of putting kind of the drug and running it through a system that would replicate what would happen if you add it to cells. But I would say human cells is the more important thing there for Mark 1. Animal testing, and then clinical trials, and you need both here, which is why I said about don't sort of just say, oh, healthy volunteers and then stop, because the next stage after that is people with the illness, sick people, patients. If you put sick people or patients, also fine, as long as you've got the idea that they have the illness and they're different from the healthy volunteers. But you need both of those to get that 3rd mark. In 1971, the deer population in an isolated forest had become too large. In order to control the deer population, the forest management team made the decision to relocate 10 wolves to the forest. The deer and wolf populations were then tracked, and the results of the programme are shown below. Okay, so we have added wolves to the forest to try and control the deer population. That's kind of code for reduce the deer population, because hopefully we know wolves would eat deer. So we've got deer population and wolf population. One of these diagrams where you've got 2 different y-axes, so just be careful when you're reading off numbers from the y-axis. Describe and explain the changes in the deer population between 1978 and 1988. So we're just focusing on this between this 10 years on the graph. Describe means what happened. So did it increase? Did it decrease? Etc. Explain is the reasons why it increased or decreased. Okay? So between 1978 and 1988, I'm just going to block off that bit of the graph so I know I'm only focusing on that bit to talk about. So the deer populations are grey with triangles. So clearly we've got an increase from 1978 up until this peak of about 300 deer, which was in 1982. And then we have a decrease down to about 50 deer in 1988. So that's the what, that's the describe. So we've got 2 points here. We've got an increase and then a decrease. So we need to explain the increase give a reason for the increase, and give a reason for the decrease. So this is really a predator-prey graph or a predator-prey relationships question. Hopefully you've kind of seen different graphs like this where you've got kind of peaks going up and down and up and down, and then the predator peak is kind of shifted to the right, but that also goes up and down and follows it. And the idea is that they go in cycles, so that's why it just asked us to look at that 10-year window. So the wolf population decreases at the start, and then the wolf population starts to increase. If the wolf population is increasing, there's more predators because they are getting more food. So the wolf population is then going to be eating the deer, which explains that decrease. When the wolf population is decreasing, which is just when our deer population is increasing, that means there's fewer predators. So that explains the increase in the deer. So there we go. So we've got a decrease in the wolf population correlates with an increase of the deer population, so we can use that to explain why there's fewer predators, therefore causing an increase. And then the decrease in the deer population correlates with an increase in the wolf population, so there's more predators who are eating more of the deer. And also, they're intrinsically linked there because the wolf population is increasing because there's more deer to eat, so they have more food. So I'm going to do like a describe, explain to make sure I've done both. And I'm going to try and use a data point in my point. So the deer population increases from 1978 to 1982 because there are fewer wolves, so less predators. Then I'm going to say the deer population decreases from 1982 to 1998, or from 300 individuals down to 50 individuals. And the explanation is because there are more wolves, because there's more food for them to eat, or because there's more deer for them to eat. Part C. If the wolves had not been introduced to the forest, suggest what would have happened to the deer population. Okay, so if there's no wolves, it's the opposite of what happens when there were lots of wolves. So when there's no wolves, there's no predators. So that means the population of the deer is going to increase. But it'll only increase to a certain point, because then the deer is going to suffer from competition. So competition within the species, so intra-specific competition, because it's said that the deer population was getting too large. And so they're going to have to compete for food with each other because there's going to be more deer than there is food available. So there'll be competition, and that will ultimately drive down the population of the deer, which is obviously what they didn't want. They didn't want the deer to starve, or they didn't want the deer to eat all the food in the habitat and then there not be anything left and then all the deer starve, which could happen. So they didn't want that to happen, which is why they introduced the wolves, which will keep the population of deer from getting too large. So 2 marks here. I'm going to say the population would increase, but then decline due to competition for food. Okay, so the mark scheme for question 22. The only thing I would say for the biotic factors is think about new pathogens and diseases. We need to be careful about saying that because often they can obviously be resistant to pathogen diseases that come around all the time. But if a new disease comes and it's less likely that they will all be resistant to it, then that can have a bigger effect on the population. Think COVID, for example. Predators, and then obviously, availability of food. You can't just say food without saying what you mean. It's about how much food there is or how easy it is for them to get. So if the amount of food, but we don't like the word amount in science, so availability of food is really important there as well. But any 2 of those get you 2 marks. And like I said, do this in this kind of pattern for B, where you do describe, explain. Data needs to be or should be quoted for the first mark. So the deer population increases from 2. So either from 1978 to 1982, which is the years, or from 25 to 30, which is the number of deer. So our explanation for why the deer population increases is that the wolf population is decreased. So there are fewer predators. So you have to link the fact that they are predators to get that mark. If you just say the wolf population decreased, but you didn't link it to the fact that, they are predators of the deer, you can't get the mark for 2. And then for deer population decreases, as wolf population increases, more deer are eaten or there are more predators. So for 3, you need to do the describe and the explain all in one to get that mark, because it's effectively a reversal or a repeat of mark point 2. And then C, without the wolves, the population of deer would have kept rising / continued to increase. So as we said, something along the lines of it would increase, or it would continue to increase, or it would continue to rise. Any wording to suggest the idea of increase in population leading to competition gets you 2 marks. So this idea of an increase gets you 1 mark, and then the idea of but they would have suffered competition for food, or there would have been competition for resources, some deer would die because they wouldn't get enough food, anything like that to get you that second mark. So the idea of an increase in the population and then an idea of competition. Any wording will get you those 2 marks because it is a suggest question. And these are where we have to take knowledge that we've been given to us in the question and kind of use our common sense or science knowledge and apply it. It's not something you will have been taught necessarily. Okay, our final question then, question 23. Sports drinks contain sugar and other salts such as sodium and potassium. A survey in a school showed that most students believe sports drinks were a healthier option than non-diet soda. The school decided to test different sports drinks and sodas being sold in their canteen to see how much sugar they contain. Okay, by sugar we mean glucose here. I've labelled the salts just because it helps me to think about in my brain what salts we're talking about, but realistically this question looks like it's going to be about glucose because it's about testing how much sugar there is in drinks, and so we're looking at glucose. Describe a method the students could have used to test the sports drinks to see how much glucose they contain. There we go. So we are testing the drinks for the amount or the presence of glucose. So they want to say how much. So we're going to need to say an actual sort of value here. We're going to try and compare them. So, but first of all, we need to describe a method. So steps in the process of how we would test the sports drinks. And then we need to think about how would we compare them to actually see how much glucose they contain. So an actual amount, not just do they contain glucose, yes or no. So this is a food test practical question. So what would you add to test for glucose? Or we'd add Benedict's. Then we need to heat it up to more than 80 degrees for 5 minutes to get it to work. And then once we've done that, we can obviously see a colour change, and that could be something we could look at to compare between the different sports drinks to see the different colours and maybe then compare how much glucose they have. But we'll come back to that towards the end. So we have to think about control variables when you're writing a method. What are you going to be keeping the same? What do we need to say they would need to measure? What would they need to make sure they kept the same as they did this test to compare the results? So the main thing is to make sure we use the same volume of sports drink for each drink we're testing and the same volume of Benedict's added to each one. So we can say they would measure the same volume of each drink, put it in test tubes, add the same volume of Benedict's to each tube, and then think of equipment. So I'm saying tubes, so we're going to need test tubes. Maybe you want to talk about pipettes. Maybe you want to talk about measuring cylinders in order to measure the volume of the different liquids you're going to need. Maybe you want to talk about water bath because we are going to need a water bath. How are we going to, we can't just say heat it up. How are we going to heat it up? We need to try and make sure there's enough description of method steps in here that someone could actually do this as a practical. That's the idea. So we need to be able to compare them. So we could compare the colours, but what if they all have sugar in? Because we know all the sports drinks probably have sugar in. There needs to be some other way we can compare how much glucose they have. But first we're going to say we're going to measure the same volume of each sports drink and we're going to put those in test tubes. You can say measure the same volume, measure the volume with a measuring cylinder. You can say how much, you can say an amount if you would like to. So say a volume, suggest a volume, that's fine. So again, measure and add the same volume of Benedict's reagents to each tube. We're not saying how much, we're just making it clear that the same amount is going, same volume, is going into each tube. Placing the tubes in the water bath at 80 degrees and leaving for 5 minutes. So that's going to get our reaction. So now we have to think about what can we do to compare them if they all turn red. That's what I would be thinking. So what we can do is we can filter, dry, and then weigh the precipitate. So it's a precipitate that is the produced that's the different colours, and it'll either be a different colour or if they all turn red, or red-orange colours because they contain glucose, then the more precipitate there is produced, then the more glucose there is. So this will also give us an actual measurement. We can weigh it, we can get a mass for the actual sort of mass of glucose and an actual number, a quantitative measurement that we can use to compare these different drinks. We'll have a look at the mark scheme because there's other things that you can say here too. So part B. The students compared the amount of sugar in sports drinks to cans of non-diet soda also being sold in their canteen. So we've got sugar in grams per bottle on the y-axis, and then we've got some sodas, and then we've got some sports drinks. So the sports drinks on the right-hand side, those 3 bars, are supposedly the healthier option according to what they interviewed when they talked to the students. So the question says, does the data support the view that sports drinks are a healthier option compared to non-diet soda? Explain your answer. So the thing here is to think about is there evidence from the data that you've got in front of you and just what's in this graph here, is there evidence to either support to say yes, sports drinks are healthier, or no, sports drinks are healthier? And if there's a bit of both, if there's any doubt, so if there's yes and no, then the answer is no. But you can mention both sides. This is kind of like an evaluate question. We're evaluating this data and we're trying to say, does it support? Does it not support? And we give evidence for both sides, and then we can kind of say yes or no. It's only 2 marks, so there isn't a lot we can say. But basically, what would the evidence be from this graph to say yes? Well, 2 out of 3 of the sports drinks have less glucose in grams per bottle than all 3 of the sodas. However, there's no evidence because the Powerade has exactly the same amount of sugar as cola. So in that case, that sort of one example shows that actually some sports drinks have exactly the same or no different amount of sugar compared to the sodas. And the Lucozade isn't that much lower, is it, either? There's not much difference between the Powerade and the Lucozade and the cola. So I would say this doesn't show that this broad statement that all sports drinks are healthier than, or sports drinks are a healthier option than sodas. And it's no, because although yes, 2 out of 3 of the sports drinks are or contain less glucose than the soda, one of them particularly has the same amount of sugar as the soda. So there's our 2 competing theories there, but we've said no overall because it does show you that there's not that much of a difference. Health advisors warn parents that the amount of sugar in drinks could be harmful to their children's health. What are the risks to health of a high sugar diet? Okay, so this is kind of one of those linked questions here. So obviously there has been a lot of news, and there's various issues around how much sugars there are in drinks available to children, or this idea that if we're selling these drinks at schools, there's a health implication. So this is kind of a health and risk factor question. So what links do we know that there are between a high sugar or a high fat or high calorie diet? What does that lead you at risk of becoming? It is a main risk factor for becoming obese or obesity. This is a 2-mark question, so what can then that lead to? Well, it's a risk factor for a lot of non-communicable diseases. An example, the main example I'm thinking of here in terms of thinking about sugar and obesity and fat around organs is diabetes type 2, specifically type 2, which is the non-communicable disease that is to do with diet and exercise and obesity being risk factor, not type 1. So that's what we're basically going to say. So a high sugar diet is a risk factor for obesity or could lead to obesity, and obesity is a risk factor for type 2 diabetes as my example. Okay, so the mark scheme for 23. So as we said, thinking about the method steps that we need to talk about how we test for glucose, we need to talk about the same volume of each drink. Do not accept amount. We can't use the word amount. We need to say what are we measuring, what sort of instructions these people are going to follow. Again, same volume of Benedicts. So you get 1 for talking about the same volume of each sports drink, then adding the Benedicts. Then we get a mark for talking about the water bath with more than 75 degrees. If you said above 75, anything above 75, you got the mark. So we said 80, so that's fine. Or the phrase boil, because that suggests going up to like 100 or near 100. So that also counts instead of saying a specific temperature. And then heat for 5 minutes, or you could say boil for 5 minutes, for example, and still got that mark. But all of that, the water bath, the temperature, and the time you leave it in, is all for 1 mark there. And then the 4th one is a method of comparison or quantification. So you don't have to talk about quantifying, getting an actual number to get the marks here. So compare colours of the precipitates. More red equals more glucose. You could have spoken about that and just talked about a colour change and comparing the different colours. You must be correct with your colour change, so talking about reds or oranges, brick reds, to indicate that glucose was present. But that could be a mark for mark 4 without having to talk about a way of quantifying it. Filter, dry and weigh the precipitate. Important here, do not forget the word dry, because obviously you don't want to weigh the solution, you would just like to weigh the solid, so it needs to be dry for you to do that. Use a colorimeter to compare the colours. Use a biosensor to look at how cloudy it is or the colour as well. So any of those got you the mark for point 4. So we said no in our analysis of what do the data show. You do not get a mark for just saying no. So if you said no, it doesn't support it, but didn't give any evidence, you do not get a mark. But we get this point that although 2 of the 3 sodas do have more sugar than the sports drinks, one of the sports drinks has the same amount of sugar as a soda. So this idea that yes, most of them were lower, but one of them has the same amount. And then for C, as we said, so high sugar diet could lead to obesity. So that's the kind of link that we know, or the condition that we know a high sugar, fat, or calorie intake diet can lead to. And then the health implications of that to get our second mark. Why is it at risk of a child's health to be obese? Well, it's because it's a risk factor for type 2 diabetes or any other named non-communicable disease. So if you said obviously it's a risk for cancer, it's a risk for heart disease, as long as you're giving an example of a non-communicable disease that obviously obesity is a risk factor for, then you can have the mark. It doesn't have to be type 2 diabetes.
Ouch! This is why in some videos I've explained scratches.